Hello students, welcome to NCERT Solutions. So today we are going to do the part 2 of what we did yesterday. So yesterday we did biological classification part 1. Today we are going to do part 2 of that chapter. So I hope I am live. Let me as well check if I am live as well. Give me 1 minute. Okay. Yes. Are we live yet? Yes, we are live, right? Awesome. Yes, today we are live. Very nice. Okay. So yesterday we did biological classification part 1. Today we are going to do part 2. So yesterday up to Protista we had done. Today we have to learn from Protista, right? We have to do fungi. So I hope all of you are ready. And yesterday's class also, I noticed that a lot of students were finding it helpful, but they were hesitant to tell at the first. Okay, but I am sure that NCERT line by line is going to help you a lot, because you're going to learn every single line with me. And learning every single line is not easy job, because most of the time what happens is students find it very hesitant to read NCERT, right? They'll be like, no ma'am, I don't want to read NCERT, it's very boring. So that time you will actually skip NCERT, or you will not take it very seriously. But this way what will happen, you will learn the chapter, at the same time you'll also learn NCERT line by line. Okay, so all of you quickly smash the like button and also subscribe to the channel. Don't do conjuicy. Okay, immediately do that as well. Now let's start with kingdom fungi, students. So kingdom fungi, so up to Protista everything is clear. Kingdom fungi, the fungi constitute a unique kingdom of heterotrophic organisms. So yesterday only we had discussed that when the organism is heterotrophic and they have a cell wall that is made up of chitin. These were the two things we discussed yesterday with your Whittaker's classification. Yes or no? So according to our Whittaker's classification, we got to know that the fungi cell wall is made up of chitin, and fungi consist of a unique kingdom where all the organisms are heterotrophic. That is, no matter what they have, resources, or they don't have resources, they will not be able to make their own food. Okay, they show a great diversity in morphology and habitat. That is what that means. They have a great variation in how they appear, right? The appearance and how, in which all regions they are present, the habitat in which they stay. You must have seen that fungi on a moist bread. How many of you have bought bread, and then after first day full excitement will be there to eat the bread, then as days pass, maybe 2 days also you'll have the excitement. On the third day you'll forget that you have left that that down pieces of the bread you have left like that, and that's when your mom will be like, look at it, it has got rotten. That is because she saw on the, now imagine this is the bread slice. Okay, this is the bread slice. On this bread slice she noticed that she started seeing some green color spots. Okay, some green color spots. Later those green color spots will turn out to be dark green. Okay, dark green, and then later it will become black in color. How that is identified? Because fungi has grown on it. The bread will absorb moisture. It is like a sponge, no? It's a sponge, so it will absorb moisture, and that moisture will lead to the rotting of the bread. Okay, I mean bread becoming bad. Then students, rotten fruits as well. All of you know that apple and all, very soon it will get rotted, right? Same way if you see tomato, you will immediately see there is a fungus growth on them. Yes or no? There is a fungus growth on them. On tomato you will notice that there is a white cast on the tomato. Okay, after some days, if you keep the tomato in the fridge for a longer time, you will notice that here all these places there will be a white color coating that is there. That is because of the fungi growth. See, fungi loves moisture. Okay, like how we love moisturizer, same way fungi loves moisture. And the moment fungi sees that, okay, wait, this area has moisture content, immediately it will go and it will start growing there. Okay, so the most common mushroom you eat and the toadstools are also fungi. So the ones that we consume, there are also some edible fungi which we consume. So white spots seen on mustard leaves are due to parasitic fungus. So if you take mustard leaf, students, this is not how it looks. One second. Yes, imagine. Okay, this is a mustard leaf. On that you will start seeing white spots. Okay, white spots on this. What is that? It is fungi. Okay, these are all fungus. Got it? So These are the white spots seen on mustard leaves are due to parasitic fungi, some unicellular fungi. So, according to Whitaker, yesterday we discussed that, ma'am, whatever is unicellular and whatever is eukaryotic should go to Protista. Then why yeast is sitting here? It is because yeast is the only unicellular fungi, and they were like, wait, this does not belong to anything like a Protista, right? It does not have any characteristics like Protista, so we will put them in fungi itself. Let it be in fungi itself, no problem, but we will keep it in fungi itself, okay? Those students who have not marked the attendance, quickly smash the like button and mark the attendance. What are you waiting for? Okay, now students, what is going to happen? White spots. Okay, white spots seen on mustard leaves are due to parasitic fungus. Okay, some unicellular fungus as yeast are used to make bread and beer. Okay, bread and beer. Now you have studied in microbes chapter, I believe, bread and beer. Yes or no? Bread and beer you would have learned, no? So, yeast, what happens, students? The story is like this. Yeast will commit suicide after the alcoholic percentage reach up to 13%. So, when we are making beer and wine ranges up to maximum up to 12% of alcohol, okay? So, that time beer is more or less, wine up to 12% it can go. 13%, after 13%, the yeast will not be able to survive. So now, when we have to make alcohol, that is 40%, 60% alcohol, like alcoholic content is there, like say your beer, rum, wine, rum, gin, like, you know, whiskey, brandy and all that, that time we have to do distillation. So there are non-distillated drinks and distillated drinks. So the non-distillated drinks will be made out of yeast. Okay, will be made out of yeast, that is up to 13%. Okay, up to 13% can be used to make all this. Okay, now other fungi causes diseases in plants, and wheat rusting causes Puccinia is important example. So Puccinia you have to remember. Some are sources of antibiotics like penicillium. Okay, penicillium. I'm not able to write. Don't know why. Okay, penicillium. Now fungi are cosmopolitan. Cosmopolitan means what? Yesterday also I wrote it. It is present everywhere. Okay, everywhere, wherever you go, fungi is present. Okay, occurs in water, in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered, students, have you ever wondered why we keep food in refrigerator? Obviously, for the main only sole reason that fungi can grow if we keep it outside, because it will lock the moisture, and moisture is more than enough for the fungi to grow. Okay, so with exceptions of yeast, which is unicellular, fungi are filamentous. So we are keeping yeast as an exception. Other fungi are filamentous. Their bodies consist of what and all? Long, slender, thread-like structures called hyphae. That is, the fungi, okay, fungi, they have long thread-like structures like this, okay, long thread-like structures like this, called hyphae. Okay, single one is called hyphae. Now, they formed a network. No, can you look here? I've made a network. A network of hyphae. Okay, a network of hyphae is called mycelium. Mycelium. Okay, a network of hyphae is called mycelium. Clear or not? So, hyphae, they are long, slender, thread-like structures are called as hyphae. And these network of hyphae, okay, is known as mycelium. Got it? Some hyphae, students, very important, are continuous tubes filled with multinucleated cytoplasm. These are called as cenocytic hyphae. So hyphae are of two types. That is, if I find like this, okay, there is one hyphae like this. It's continuous, okay, continuous and multinucleated, like this. Okay, this is one hyphae. This hyphae is called as cenocytic hyphae, or I can also call this aseptate hyphae. Aseptate hyphae. What is the meaning of that? That is, see, if I draw this, you will understand. See, this is another hyphae. Okay, this is another hyphae. Dot, 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 dot. Okay. See, in between like this barriers are there, right? Now, if we both stay in the same layout, okay, we both stay in the same layout. You have built a home, I have built a home. What is the first thing we do? We put a fence, we put a proper covering, right? That this is my house, that is your house, so you have the privacy, when I have the privacy. Same way, this hyphae also has something called as septas or boundaries, okay? That is called, so whenever the septas are present, no, we will call it as septate hyphae, okay, septate hyphae. Now septa is not there, that breaking is not there, chumma one hyphae is there with so many nucleus, then that we call it aseptate or cenocytic hyphae. Clear or not? So only if you understand septate, you will be able to understand aseptate. Okay. Now see here, they have already mentioned that. Others have septae or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. So we have already discussed that the cell wall is made up of chitin. Now they are telling that it is made up of chitin as well as polysaccharides. Okay, sugar molecules. Now most fungi are heterotrophic and absorb soluble organic matter from the dead substrate, and hence are called as saprophytic. That is, they feed on dead and decayed organism and they take the organic materials from them. Okay. Now those that depend on living plants and animals are called as parasites. Obviously you understood, no? Something is waiting, like in the sense I'm like, okay, I am very dependent on this person. Whatever this person's nutrition is there, I will take it. I'll become a parasite. Okay, I'll not become a parasite. Example, okay, I'll become a parasite. Now imagine like two people, okay, they're living in very much harmony, they're happy, they're like, okay, I will cook food for you, you give me shelter. This will become a symbiotic relationship, which we see in lichens. Lichens is actually an association with fungi and algae. Okay, fungi and algae. Now algae will give food for the fungi and fungi will give shelter for the algae. So many of you would have seen lichen in your real life, but you wouldn't have recognized then. But I'll tell you how it looks. Okay, with lots of higher plants as mycorrhiza. Mycorrhiza, students, you see same this association is there, that is seen with higher plants. Okay, higher plants. Higher plants. Clear? Clear? Moving on. Now reproduction in fungi can take place by vegetative means. Okay, by vegetative means, that is fragmentation, fission, budding. Obviously reproduction has to take place. Now in Monera we understood that reproduction happens by the help of fission. Here the telling it can happen with the help of fragmentation, fission and budding. That is, if there is a fragment, okay, this is the fragment, it will go into pieces, each of it is a fragment and each of it can give rise to a new individual. Okay, each of it can give rise to a new individual. Fission is there where two pieces are there that will get separated, that will be called as fission. Now budding means what? On one, another one is growing, a daughter is growing on the parent, after some time this will separate and it will start growing by itself. Okay, budding. Now asexual reproduction is by spores. This is very important. Asexual reproduction. So students, if, like most of the time what happens, students get confused with vegetative and asexual. Vegetative means their own parts of the body is reproducing, that is able to make another new individual. Now asexual means what? Variation is not happening. Okay, mother, father, like male and female fusion is not happening, but still they are reproducing. Okay, asexual reproduction is by spores, that is called as conidia. So students, this is the only confusing part in fungi, where you have to know which are the sexual spores, which are the asexual spores. So here asexual spores are conidia or sporangiospores, okay, or zoospores, okay. These three are the spores of your asexual reproduction. Now the sexual reproduction is by oospores, ascospores, basidiospores. Can you see here it is or here it is comma. That means everything is separate. Okay, the various spores are produced in distant structures called fruiting body. So the spores will always be inside a fruiting body. So now this is the fruiting body. Okay, one second. Yes, this is the fruiting body. Inside this spores will be there. Okay, inside this spores will be there. Now the sexual cycle involves following three steps. Fusion of protoplasm between two motile, okay, between two motile and non-motile gametes are called as plasmogamy. What are they telling? Fusion of protoplasm. Fusion of protoplasm between two motile and two non-motile gametes. Okay, two motile means both are moving. Okay, let's say this one also has a flagella, this one also has a flagella, these two fuse together, or two non-motile fuse together also, it will happen. What will happen? So when these both contain, what is that? Protoplasm. Okay, protoplasm means cytoplasm plus nucleus. Okay, is it visible, student? Yeah, no, visible, no? Okay, my glasses are actually giving reflection, no? Okay, I'm just removing the glasses. Okay, so protoplasm, one second, I'll just clean the glasses. I think it should be better now. Okay. Yes. Fusion of two nuclei of the due to two. So first fusion of protoplasm happened. That is, protoplasm means cytoplasm plus, okay, cytoplasm plus nucleus. Okay, always remember this. Many students don't know this. It's okay to not know, but learn it now. Protoplasm means cytoplasm plus nucleus. Okay, so when two cells are fusing together, what happens? The cytoplasm mix as well as the nucleus will come together. Okay, now fusion of two nuclei is called as karyogamy. All of you know karyo word is always based on nucleus. Now meiosis in zygote resulting in haploid spores. That is the last stage. Meiosis will happen, zygote will result in haploid spores. Okay, so here I just want to write these things. Okay, so students, very simple. Vegetative reproduction. Okay, vegetative reproduction. Okay, vegetative reproduction happens by three techniques. Okay, three techniques. What are these three techniques? Fission, fragmentation, okay, and budding. Okay, and budding. Okay, now this is what has happened. Now if I go to asexual spores, okay, asexual spores, they can be conidia, okay, zoospores, or sporangiospore. Now I'll tell you one thing, conidia is the only one that grows exogenously. Exogenously means, let's say this is the fruiting body, okay, this conidia will grow like this. Conidia will grow outside, okay. This is called as exogenous, okay, this is called as exogenous. Got it? So conidia is exogenous, grows exogenous. Yes. Now sexual reproduction. Sexual reproduction. Okay, sexual reproduction. Sexual reproduction also happens with the help of three spores. Okay, first one is called as you. Okay, oospores, ascospores, basidiospores. Okay, basidiospores. So from the term ascospores and basidiospores, you should be able to understand this is going to fall under your Ascomycetes and Basidiomycetes. Okay, at least that much you should be able to understand. Oospores, ascospores, and basidiospores. Now students, they are telling that imagine two cells are there. This is one cell. Okay, let's say this cell is motile. Okay, this is one cell which is non-motile, or both are motile. Now they both will fuse together. First thing that is going to fuse with them is going to be the protoplasm. Okay, protoplasm. This protoplasm fusing is called as plasmogamy. I write it here. I don't think you will be able to see, no, right behind my head. Okay, plasmogamy. Can you see it? Okay, now after plasmogamy, I'm taking it here. Now the two nucleus, this two nucleus has to fuse together. This nucleus fusing will be called as karyogamy. Okay, karyogamy. Now after karyogamy, obviously meiosis will happen. Meiosis will happen to form zygote. These are the three steps of your sexual reproduction. Okay, three steps of your sexual reproduction is going to be plasmogamy, karyogamy, and zygote formation with the help of meiosis. Clear? Okay, moving on. Yes, when a fungus reproduces sexually, okay, when a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells. That is, n plus n will give you 2n, right? So two haploid cells will give rise to a two diploid cells. Okay, two diploid cells. Now, however, in other fungi, Ascomycetes and Basidiomycetes, an intervening stage is called as the dikaryotic stage. That is what is going to happen, students. We all understood in the first stages, plasmogamy is going to happen. Both of them will come together. Okay, both of them will come together. What did I tell you? Second stage is going to be where the nucleus is going to come together. But in some, no, what is going to happen? This two nucleus is going to stay like that only. Okay, this two nucleus is going to stay like that only. They will be like, what? This is an intermediating stage, which is called as the dikaryon stage. Okay, dikaryon stage. That is, both the nucleus is not fused with each other. Instead, they are remaining like that, and that will form an intermediate condition called as the dikaryon stage, or the phase is called as dikaryophase. Dikaryophase. Okay, later the parental nuclei fuse and the cells become diploid. So students, very simple, in Ascomycetes and in Basidiomycetes, what is going to happen? There is going to be a dikaryose phase where both the nucleuses will come together, but they will not fuse with each other. They will remain as that. So that is why di means what? Two caryon stage. Caryon means whenever the word caryon come, understand it is something to do with nucleus. So dikaryon phase of the fungus. Later the parental nuclei fuse and the cells become diploid. The fungi forms fruiting bodies in which reduction division occurs. Okay, in which reduction division occurs, leading to the formation of haploid spores. Okay, formation of haploid spores. Okay, the morphology of the mycelium, mode of spore formation, and fruiting bodies are the basis of classification of the fungi. That is, morphology of the mycelium, how the mycelium is it septate, aseptate, what type it is, mode of the spore formation. Okay, what mode it is it sexual mode, is it asexual mode, if sexual and asexual is there, what is the type that they are forming. Then fruiting bodies, like if they are like, you know, different things are there, like basidiocarp, ascocarp and all that. So depending on that also the classification is done. Okay, so the first ever classification of your fungi is called as Phycomycetes. What is it called? Phycomycetes. Okay, look here. Members of Phycomycetes are found in aquatic habitats. Okay, aquatic habitats and on decaying woods. They are seen on decaying woods, in moist and damp places as well. Okay, or as obligate parasites on plants. So these people are what and all? They are found in aquatic habitats, on decaying woods. Okay, on decaying woods as well as on plants as well. Okay, 1 second. Yeah. Plants as well, they are found in damp places or even in obligate parasite. Okay, the mycelium is aseptate and cenocytic. What is aseptate and cenocytic? I already told you. They have no septa. Okay, they have no septa or no cross walls. Okay, in other words, I can tell no cross walls. Got it? And cenocytic, as I told you, multinucleated long strands. Okay, thread-like structure. Asexual reproduction takes place by zoospores. Zoospores, that are motile one, or it can be non-motile. Students, for non-motile, we can also use the word aplanospores. Okay, you can also use the word aplanospores. These spores are endogenously produced in sporangium. That is, endogenously means what? Inside. Okay, now this is the fruiting body. Let's say this is the fruiting. I think I should draw a little more up. This is the fruiting body. This might look like a balloon, but it's not. Okay, this is the fruiting body. So they are telling that this one is growing inside. Okay, endogenously, inside. Students, this is one reason why fungi is so strong. Okay, it's because of the formation of spores. So when the condition gets suitable, they will form the fruiting bodies. When the condition gets unfavorable, they'll rupture open the fruiting bodies and the spores will migrate. Okay, these spores are endogenously produced in the sporangium. Endogenously means inside. Okay, in the sporangium, this can be called as sporangium. A zygospore is formed by the fusion of two gametes. These two gametes are similar in morphology. Now, depending on what type of gametes are there. Okay, what type of gametes are there? One second. Okay, what type of gametes are there? Isogamous is there, anisogamous is there, oogamous is there. So some common examples are mucor, rhizopus, albigo. Okay, albigo. Now remember students, here they are telling example, you have to remember like this. Mars, you can remember Mar, that is M stands for mucor, A stands for your albigo, and R stands for rhizopus. Remember like this, Phycomycetes, Mar. See, look at this. See, in the potato, all of you have seen, huh? Potato, yes or no? Yes, in potato, all of you would have seen like this, okay? Like, you cannot see it with your naked eye, but you would have seen this green color spots. If I see the microscopic of this green color spots, it will look like this. See? This will be the sporangiophore, okay? This is be the stolon, that is where they grow, rhizoids, and this sporangiospores will open up and the spores will be there. So, this will be called as the sporangium, okay? Sporangiophore is the one that is holding, the stalk that is holding, and this will be the sporangium, okay? This will be the sporangium. Got it? Inside the spores will be there. The spores will get scattered. Spores will get scattered. Now, what did I tell? Phycomycetes. Mycetes. Okay, first thing you have to remember, students, asexual spores. Okay, asexual spores are with the help of zoospores. Okay, asexual spores are with the help of zoospores, and they are endogenously formed. Okay, endogenously formed. Then here they have told about that is sexual spores. No, in sexual spores they use some words. What are those words? First word that they used was isogamous. Isogamous. Isogamous means what? That is, imagine this is one spore. Okay, this is one more spore. That is, both are same size, shape, and motility. Everything is same. Twins, like twin students. Okay, yes, twin students, same size, shape, motility, everything, isogamous. Okay, that is, they will have the similar size and shape, and if both are motile, both will be motile. If both are non-motile, both will be non-motile. That kind of spores are called as isogamous. Then the next one is called as anisogamous. Anisogamous. That is what? One will be big, okay, one will be cutie, small. Okay, one will be big, one will be small. Got it? This is called as anisogamous. But here also remember, both will be, okay, different size, but same mobility. That is same motility. That is, if both are moving, both will be moving, or if one is moving, it will not be like one is moving, one is not moving. Both will be having flagella, or both will not have flagella. Okay. Last one is, students, oogamous. Oogamous is just like in humans. Okay. Oogamous is just like humans. That is where you have one large female and one small male. Okay. Here size will be different. Even motility will be different. Got it? Motility also different. So this is what I wanted to tell. Now Ascomycetes. Commonly known as sac fungi, the Ascomycetes are mostly multicellular, that is penicillium, or rarely unicellular like yeast. Okay, Sarcomycetes, they are saprophytic, decomposers, parasitic, or coprophilous. Very important. They grow on cow dung. Okay, they grow on cow dung. So what they are telling, commonly known as sac fungi. Sac fungi. Why you'll get to know why they are sac fungi, and they are mostly multicellular. Example, penicillium, or rarely unicellular like yeast. Okay, both extremes are there in this kingdom. I mean in this class, where you have multicellular penicillium and then you have unicellular yeast. Okay. They are saprophytic, decomposers, parasitic and coprophilous. Mycelium, very important here, mycelium is branched and septate. Students, here we forgot to write that Phycomycetes is the only one that has an aseptate, okay, aseptate mycelium. Aseptate cenocytic mycelium. Okay. Yes, Jenny, I will be completing the chapter today. Okay. Branched. This is starting from here. It's going to be branched and septate. The asexual spores are conidia, produced exogenously. I already told you conidia is exogenously going to form. That means it's going to form outside, not inside. Okay. On a special mycelium called as conidiophores. Okay, conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores. Okay, sexual spores are called ascospores. As I told you, Ascomycetes will have ascospores. Basidiomycetes will have basidiospore. Understood? So here they are telling that the septate was branched and septate. The asexual spores are conidia, which are produced outside exogenously in a special mycelium called as conidiophores. Okay, conidia on germination will produce mycelium. Sexual spores are called as ascospores, which are produced endogenously inside a sac-like asci. This is how the term sac-like fungi came. Okay, sac-like asci. These asci are arranged in different types of fruiting bodies called ascocarp. Okay, so the fruiting bodies are called as ascocarp. Now, some examples are aspergillus, claviceps, and neurospora. Neurospora is used extensively for your genetic engineering. Okay, genetic works, biochemical works, and all these are used. At the same time, students, there is murals and truffles which are edible. This is mainly consumed in places like Korea for ramen, or if you know ramen noodles, it is mainly consumed. Okay, this murals will be something like this. Okay, murals will be sliced in front of them, and that has a special cost by itself. Okay, for that add-on and all, you have to pay more money. Okay, that is murals and truffles, which are I would say not as common as mushrooms, little more rare. Okay, little more rare. Yes. Okay, yes. Since I'll just get some water, till that you just please read this. Yes, students, I'm back. I just finished the class, so my voice is tired. Okay, so students, as I told you, Ascomycetes. Okay, Ascomycetes, they are sac-like fungi. Okay, and they're mostly multicellular. Penicillium is there, and then rarely unicellular. Yeast is there. So at one corner you have a multicellular organism, in another corner you have a unicellular organism, and they are saprophytic, decomposers, parasitic, and coprophilous. That is, they are found on cow dung as well. Okay, now these people are branched and septate. Asexual spores are conidia, produced exogenously outside, okay, on a special mycelium called conidiophores. Now conidia on germination produces mycelium. Okay, sexual spores are called ascospores, which are produced endogenously in a sac-like fungi called as asci. Okay, asci are arranged in a different types of fruiting body called ascocarp. Some examples are Aspergillus, Calviceps, and Neurospora. In that, Neurospora is used for all genetic works, and then there are also edible ones like murals and truffles. Okay, murals and truffles. So here we will write Ascomycetes. Okay, Ascomycetes. What did we study? First examples we will write. We have Penicillium. Okay, Penicillium, which is multicellular. Okay, and then we have yeast, which is unicellular. So two extremes. Okay, then most important, they have septate and branched mycelium. Okay, branched hyphae. And these people, okay, they have asexual spores called conidia. Okay, conidia. Is the handwriting visible? Color is okay? Yes, okay. Branched conidia. I think I'll write it with white as well. Conidia. This conidia is present on special mycelium. Okay, special mycelium called as conidiophores. Okay, conidiophores. Okay, conidiophores are the ones on the which it will be present. Now, sexual. This will be formed exogenously. Okay, exogenously, no doubt. Okay. Now, sexual spores are, students, obviously they are called as ascospores. Okay, ascospores. These ascospores are present in a sac-like, okay, sac-like structure called as ascocarp. Okay, asci. First, the sac is called asci. First, we'll write that. Okay, asci. Then they are present on fruiting bodies. Okay, fruiting bodies called ascocarp. Okay, got it. Ascocarp. Done. So many ants are playing. Okay, ascocarp. So in this fruiting bodies, these people will be present. Like this. Let's say this is the normal. This is how usually we draw fruiting bodies. Okay, don't think why this ma'am for everything she's drawing same thing. This is how usually we draw fruiting bodies, or we draw like this. Okay, one circle. Again we draw like this, one circle. Inside this it will be present. It will be present. Okay, that's how it will be present. Now example, you have Aspergillus. Okay, Aspergillus, Neurospora. Okay, you can remember murals, truffles. Okay, in that these two people are edible. Done. Okay, done. Yes. Okay, next is Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, the one that we consume. bracket fungi, puffballs. Okay, these are the types of Basidiomycetes. They grow in soil, on logs, on tree stumps, and in living plant bodies as parasites. Okay, example, rust and smuts. The mycelium here also is branched. I told you the only unbranched or aseptate mycelium is seen in Phycomycetes. Okay, the mycelium is branched and septate. The asexual spores are generally not found. Okay, but vegetative reproduction is by fragmentation is the most common one. So vegetative reproduction here is by fragmentation. Asexual spores are not known. Okay, they are not found. Now the sex organs are absent, but here students you will see something different. Okay, sex organs are absent. That means what? They do not have sex organs. Okay, no plasmogamy, none of that. For that, sex organs are not there. Okay, but look here, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. Got it, all of you? So they're telling that even though no sex cells are there, we are going to use the vegetative cells. Okay, vegetative cells or somatic cells. Okay, that means the cells which will not show any variation. They are the normal cells, right? With them only we are going to do plasmogamy. Plasmogamy. Okay, fine. After that, what? Okay, after that, what they're going to do, students? Once the plasmogamy is done, the resultant is going to be a dikaryotic stage. Okay, dikaryotic stage. We already understood that, no? In Ascomycetes and Basidiomycetes, it's going to be a dikaryon stage. Now, this dikaryotic stage will give rise to what? Basidium. Okay, this will give rise to something called as Basidium. Okay, Basidium. This basidium will form 4 basidiospores. What will form? 4 basidiospores. Got it? 4 basidiospores. The basidiospores are exogenous, produced on basidium. Okay, so the ones, this basidiospores, no, they're formed. Let's say this is the basidium. On the top of this, they are formed. Okay, on top of this structure, they are formed. Got it? Now the basidia are arranged in fruiting bodies called basidiocarp. So now this basidium or basidia are arranged inside fruiting bodies called basidiocarp. So whenever carp word come, think about it as fruiting bodies. Okay. There also we saw ascocarp. Here we saw basidiocarp. Got it? Okay. So some common members are agaricus, mushroom, Eusteligo and Puccinia, that is mushroom, smut and rust fungus. Okay, rust fungus. So here what and all we saw, most common one is mushroom, bracket fungi of puffball. They grow in soil, logs, tree stumps. Plant bodies are parasitic. Okay, mycelium is branched and septate. The asexual spores are generally not found. So here only thing that you have to worry is about vegetative. But here sex organs also are not present. But the somatic cells are going to fuse together and do plasmogamy. That plasmogamy will give rise to dikaryon. That dikaryon will form a basidium stage. On the basidium there will be 4 basidiocarp that will be forming. That 4 basidiocarp will be formed exogenously, right? Outside it will be formed, and then what is going to happen, students? The resultant structure will undergo karyogamy. So here look here, the resultant structure is a dikaryotic stage which ultimately give rise to basidium. Karyogamy and meiosis will take place in the basidium. Okay, everything will happen in the basidium, producing 4 basidiospores. Now this basidiospores will be formed exogenously, produced on the basidium. This basidium will be kept inside a fruiting body or arranged in fruiting bodies called basidiocarp. Got it? So here let's write that. Okay, Basidiomycetes. Okay, mushroom, bracket fungi. Here what is going to happen, students? Here you have to remember that first thing is they do not have asexual spores, not found. Like it could be found anytime, but now as of now it is not found. Vegetative reproduction, no, vegetative reproduction is by fragmentation. Okay, is by fragmentation. There is one fragment is there. Okay, this is branched and septate. Okay, they will be broken into pieces. This each piece can give rise to a completely new individual. Okay, completely new individual. Okay, now here, yeah, I write it here. They are septate and branched. Septate and branched hyphae they have. Vegetative reproduction is fragmentation. Now, sex organs, okay, are absent, not found. No, they are absent. Okay, they are absent. But what is the solution we have? What is the solution we have? For everything we have solution, right? Okay, for the solution is that the somatic cells, somatic or vegetative cell, okay, somatic or vegetative cells will undergo plasmogamy. Okay, will undergo plasmogamy. Now this plasmogamy, what students? Fusion of protoplasm. No, so this plasmogamy will give rise to a dikaryon stage. It will give rise to dikaryon stage. Okay, got it? Dikaryon stage. Now this dikaryon stage, okay, this dikaryon stage will give rise to basidium. Okay, this dikaryon stage will give rise to basidium. Okay, basidium, clear? Dikaryon stage will give rise to basidium. Now this basidium, okay, this basidium will grow basidiospores. Okay, basidiospores. And how will they grow? How will they grow? They will grow exogenously. So let's say this is how the basidium looks like. Okay, this is how the basidium looks like. On the top, like this, exogenously it will be growing. Okay, exogenously it will be growing. I will erase one because there's only 4. Okay, basidium, exogenously it will be growing, giving rise to basidiospores. Now remember that this basidium will be arranged. Okay, if I write here, is it visible? Okay, arranged in fruiting bodies. Arranged in fruiting bodies, called as what? Basidiocarp. Basidiocarp. Got it? Okay, understood? So what is going to happen, students? Somatic or vegetative cells will fuse together, and that is going to be called as plasmogamy. Plasmogamy means what? Cytoplasm plus the nucleus will fuse. Okay, that is when this plasmogamy will happen. That plasmogamy can happen with sex organs, but here sex organs are absent. So this plasmogamy will fuse and they will form a dikaryon stage. Dikaryon stage means what? Both the nucleus will remain just like that. They will not move. Okay, they will not fuse together. They will just remain like that. That is called as dikaryon stage or dikaryophase. Now, this dikaryon stage will lead to a formation of basidium, and that basidium will be able to form basidiospores. Okay, 4 basidiospores it will form. This 4 basidiospores will be there. Now, this basidium will get arranged in fruiting bodies called basidiocarp. Clear? Okay, now example you can remember: mushroom, bracket fungi, Usteligo, many other. Usteligo. Oh. Okay, Usteligo, Puccinia. No, Usteligo you can remember. Puccinia is not there. Okay, Puccinia you can write. Yeah, Puccinia also you can write. But if you can remember, okay, otherwise go for mushroom and bracket fungi, easy to remember. Ready? Done? Yes, okay, done. Yes, now next, last fungi. Okay, last fungi. This fungi has actually nothing, students. This fungi has actually nothing. Commonly known as imperfect fungi, because only the asexual or the vegetative phase of this fungi is known. That is, we should have asexual, vegetative and sexual phase. Here what is known? Only asexual or vegetative phase is known. That also odd. When the sexual forms of these fungi were discovered, they were found or moved into classes they rightly belong. So what happened with this fungi is, this fungi is called imperfect fungi, mainly because whenever something was found about this fungi, no students, they were put into another place. Okay, they were put into another place. See, when they found the sexual phase, they were migrated somewhere else. When the asexual phase was known, they were put somewhere. So that confusion was always there with this Deuteromycetes. Okay, it is also possible that the asexual and the vegetative stage have given one name and the sexual stage another, and placed under another class. So I told you, when the sexual phase was known, they were put into another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. One perfect sexual stage of the members of Deuteromycetes were discovered, they were often moved to Ascomycetes. So whenever sexual phase of Deuteromycetes was found, they were pushed into Ascomycetes, telling that you do not belong here, you go that side. Okay, and Basidiomycetes. Now, the Deuteromycetes reproduce only by asexual spores. Okay, so Deuteromycetes is only produced by asexual spores, and the spores are called as conidia. Again, remember, conidia is exogenous. Okay, exogenous. Now, the mycelium is septate and branched. Okay, mycelium is septate and branched. Some members are saprophytic or parasitic, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, see, you cannot remember this, at least remember Trichoderma and Alternaria. Okay, Coleotrichum, if you cannot remember, don't remember, but at least remember 80. Okay, Alternaria and Trichoderma. Okay, done. This is done. So here, as I told you, only one point you have to remember, they are called as imperfect fungi. Why are they called imperfect fungi? Because only, okay, only asexual, one second, only asexual phases known. Asexual phase is known, that is conidia. Okay, conidia. This is only asexual phase that is known. Okay, next topic is, students, viroids, prions and lichens. Okay, so this did not, the problem was, you know, there are some people, no, in life where we cannot call them friends, we cannot call them enemies, like they are in the middle stage. Same way these people are like that, we cannot put them into any kingdom, so we put them separately, right? We just, nowadays people call, no, many what is that, relationships are called different, okay, different thing. Yes, Roshni, very good. Different things are called, no, situationship, something, okay, some ship and all that, it's called. So like that only, everything has its own name. Same way, these people are not going anywhere else, they were not belonging anywhere else. So then people were like, okay, let's not take tension, let's do one thing, let's just put them separately. Okay, let's just learn about them separately. That is your virus. Okay, viroids, prions and lichens was learned separately, not under any kingdom. So in the 5 kingdom classification of Whitaker, there is no mention of lichens and some acellular organisms like virus, viroids and prions, because students, now also the question still goes, the argument about if virus is living or non-living is still happening. Then how can Whitaker put them anywhere? Right, Whitaker will be like, wait, how can I put this fellow somewhere? I don't even know if this fellow is living or non-living. Okay, so all of us who have suffered the ill effects of common cold, just now I taught human health and diseases and came, the virus is called rhinovirus. Okay, rhinovirus, and flu is caused. Okay, known what effects virus can have on us. even if we do not associate it with our conditions, virus did not find a place in the classification since they are not considered truly living. Students, that's why virus does not like humans, because we did not give them a proper class. Okay, we did not give them a proper kingdom. So virus always keeps affecting us, especially rhinovirus, common cold. I hope all of you know that every then and now we all get common cold. Yes or no? The virus are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. The main problem with virus is it is only going to act living when it finds a host. If no host is there, then it is non-living, because it will have a protein code and it will have a genetic material. If it is not, like, if it does not find a host, then who is going to, like, you know, where is the connection going to come, right? Where is the connection going to come? So that is the problem here with virus. So here you look, students, virus structure. This one is a bacteriophage. Okay, bacteriophage phase. This is a virus, okay, that infects bacteria. Okay, this is a virus that infects bacteria. That is, they have a head, they have a collar, and this is the sheath, and then they have these legs, okay, tail like fibers like this. Now what they will do is, imagine, let's say this is the bacteria that we have to affect. Let's say this is an E. coli. Okay, this is an E. coli. Okay, what this fellow will do, no, this is the bacteriophage, no, it will go like this. This is the bacteriophage. Like this, it will affect. Okay, affect andre, it will not just hold and pinch or something like that. Okay, it will release the genetic material inside. It will release its genetic material inside. It could be a double-stranded DNA, it could be a single-stranded RNA, it could be any of it. It will just release the genetic material inside. Now this genetic material, what it's going to do? It's going to change the machinery of this cell. Okay, it is going to change everything about the cell. Students, imagine an uninvited guest come to your house and they start changing everything in your house. They're like, hey, this interior is not good, this is not where the kitchen should look like, how this is, how this is. Same way this virus will do the same thing. It will get into the host body. Okay, it will get into the host body. It will change the entire machinery of the host body. It will be like, wait, I will tell you what to do in life, and it will change the whole thing. Okay, it will change the whole thing. So here you can see, here this is this structure, no. Now if I take the outside, this portion of it, you will notice that this one, these each single individual, like this petals are there, no, these are called as capsomeres. Many capsomeres. Okay, many capsomeres together forms a capsid. So this is the protein code. Why the protein code is there? To protect the genetic material. So genetic material is everything. If the genetic material does not enter the host, then the virus is not at all a problematic fellow. But if the genetic material enters the host, it will manipulate the host. It will tell host what to do, what not to do. Okay, so even our body, you know, whenever see, HIV is AIDS, the ones that cause AIDS. Okay, human immunodeficiency virus. This HIV, no, students, it will completely manipulate the host. Okay, manipulate the host machinery. Now what is going to happen now? We are going to work according to virus. We are not going to work according to our central dogma. So that is why virus is very, very dangerous. So here you can see, one second, yeah. Only they infect a cell, they take over the machinery of the host cell. This is exactly what I told. Okay. Normally in our body, what happens? DNA is there. DNA will become mRNA. Okay, DNA will become mRNA. mRNA will become protein. Correct? Same thing will happen. But the problem is, we will start forming RNA for the virus. Okay, we will start forming proteins for the virus. Viral proteins will start forming. By this, what is going to happen, you tell me? The entire body will have virus and it will kill, okay, it will harm our cells. So this is what is going to happen. Okay, now see, it will replicate, killing the host. Would you call virus living or non-living? This argument still continues because they have a lot of characteristics like living, like they can replicate, it's a living character, but they also have characters like non-living, right? They do not grow or they do not reproduce. Like they do reproduce, I mean they replicate. Okay, they do not reproduce, they replicate. So many non-living and living characters are a mix. Okay, now virus means venom or poisonous fluid. Okay, so Dmitry Ivanovsky in 1892 recognized certain microbes as causal organisms. Okay, causal means something that causes any disease of mosaic disease of tobacco mosaic virus. Okay, now these were found to be smaller than bacteria because they pass through the bacterial proof filter. Students, we all do chai, no? In chai we filter it out, no? What happens if there is tea leaf? Tea leaf will get stuck in the this one, no? Same way here he used a bacterial filter proof to understand if the disease caused on that mosaic, okay, on tobacco was bacteria, but he noticed that it is smaller than bacteria and it was passing through this tubes, in this filter. So he was like, wait a minute, this is something else. Then Bejernik in 1898 demonstrated that the extract of the infected plants of tobacco had caused infection in healthy plants. That is, when they took the fluid or when they took the content of the infected plant and they tested it on healthy plant, they noticed that healthy plant was getting affected. Then they were like, wait a minute, this is some new pathogen by itself, and they called this fluid as Contagium vivium fluidum. Okay, that is infectious living fluid. Then W.M. Stanley in 1935 showed that virus could be crystallized, and crystals consist largely of proteins. Yes or no? Outer coat is protein. Okay, outer coat is protein only. This is protein. I am drawing bacteriophage because you saw the bacteriophage. Okay, this is the protein. Okay, this is protein only. This is protein. So largely it is protein. Inside this genetic material is going to be, could be double stranded or something. Okay, the genetic material. So they told, Stanley told that, wait, they are something that has rich protein. They are inert outside their specific host cell. Virus are obligate parasites. Okay, virus are obligate parasites. In addition to proteins, virus also contain genetic material. That is the main problem. Problem is this fellow. Okay, yes, exactly, Aditi, yeah. The exact problem is that, students, that this fellow has genetic material. Got it? This fellow having genetic material is the biggest problem. Okay, it can be DNA or RNA. Exactly, it could be single stranded or double stranded. Okay, and now they have also found a virus which has both DNA and RNA. I think it's called leucovirus or something. Okay, that could be neither DNA or RNA. No virus contains both RNA and DNA is given here, but if you check, I checked recently, I think it is leuco or something. Students, just check and tell me what is a virus that has both DNA and RNA. A virus is a nucleoprotein. Nucleoprotein means what? That is, nucleic acids are there. Okay, nucleic acids are there and they are protected by a protein coat. Now that protein coat, you have to remember one thing, that protein coat, one second, that protein, where am I? Yeah, that protein coat is made up of capsomeres. That capsomeres fuse together to form capsids. Okay, and inside this capsids the protein will be there. Got it? A virus is a nucleoprotein and the genetic material is infectious. In general, virus that infects plants have a single stranded RNA, and virus that infects animals have neither single or double stranded. Then only they'll be able to manipulate the host this. Okay, then only they'll be able to manipulate it. So they're telling generally, not every time, it's not necessary. The ones that infect plants, leuco only, no? Okay, leucovirus only. That's what I had read it. Okay, plants. The virus that infects plants will have single stranded. Okay, single stranded RNA. Okay, the ones that affect animals will have ssRNA. Okay, they could have ssRNA, or they could have dsRNA, that is double stranded RNA, or they could have ssDNA. I mean, the ssDNA is not there. Okay, they will have dsDNA. Okay, dsDNA. Got it? So this is what they're telling. So these people can manipulate the host. Okay, they can manipulate the host. If they have only single stranded, then they cannot affect us, no, because again we have double stranded. So They can have single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, virus that infects bacteria, are usually double-stranded DNA virus. The protein coat called capsid, made up of small subunits called capsomeres, which I already discussed now. Capsomeres, capsids, now protects the nucleic acid. That is the main job of it, to protect the nucleic acid. Now these capsomeres are arranged in helical or polyhedral geometric forms. Okay, geometric forms. Virus cause diseases like mumps, smallpox, herpes, influenza. Okay, AIDS in humans also are caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing, vein clearing, okay, dwarfing, and stunted growth. My god, all this is the symptoms in plants. Okay, you would have noticed, students, leaves rolling, the leaves, veins will be disappearing, all that. Okay, all that is the symptoms in plants. Very like symptoms that you would have observed, okay, in plants which are affected by virus. Now viroids. Let's see what is viroids. So in 1971, D.O. Denner discovered a new infectious agent that was smaller than virus and caused potato spindle tuber disease. What did they cause? Potato spindle tuber disease. It was found to be free RNA. Oh, so viroids, see, that's what I'm trying to say. Viroids, okay, tobacco mosaic virus, Aditi. Tobacco mosaic virus. Okay, so students, a new infectious agent which is smaller than virus. So virus is there, smaller than that, viroids is there. Okay, now viroids have what? Okay, viroids have what? Viroids have only free RNA. Okay, only free RNA. Free RNA. So no protein coat is there. It lacked protein coat that is found in virus. Okay, hence the name viroids came. The DNA of the viroid was of low molecular weight. So the ones, the RNA, the RNA that was there had low molecular weight. Okay, low molecular weight. Now next is prions. Okay, prions. Now prions are, in modern medicine, certain infectious neurological disease were found to be transmitted. This is very crazy to understand that they were transmitted by an agent consisting of abnormally folded protein. Okay, abnormally folded protein. That means even though we thought that protein cannot cause an infection, they found that abnormally folded proteins, okay, these folded proteins also caused infection. Okay, folded protein also caused infection, but very selective infections. Okay, not like chumma, whenever they walk around they'll cause an infection. Okay, normal, the ones that they were possibly able to move. Okay, students, likes have an increase. What happened? Have you forgotten to like? Okay, yes, 1 second. Okay, so the agent was similar in size of the virus. These agents were called as prions. That is, with the help of folded proteins, they started making diseases. The most notable disease caused by prions are bovine spongiform encephalopathy, that is BSC, commonly known as mad cow disease in cattle. Okay, this is a very painful disease for the cattle, students, because the cattle actually suffer a lot when this disease happens. Okay, mad cow disease. And in it, analogous variant is the CR Jacob. Okay, I don't know why I told cry. It's a very difficult name, crucified Jacob disease. Okay, which you don't have to know, it's some other language name. So it is Jacob disease, CJD disease in humans. Okay, so that is analogous. That means analogous variant, homologous, analogous variant. No, analogous variant is your Jacob disease. So this prions are folded proteins that causes diseases. Okay, that has the capability to cause disease, and the disease they cause is mad cow disease. Mad cow disease. Okay, got it? Yes, cool. Okay. Okay, we'll resume after 5 minutes. Okay. Okay, we'll resume back at 5 p.m. students. Oh, 5 p.m. is a lot. 1 second. We'll resume back. Four 40. Okay, 10 minutes. Hello students. Yes, so let's continue. So we did viroids, virus and prions, right? What did we learn out of it? So we learned that virus, okay, virus has two things, okay, a protein code. Okay, 1 second students. Okay, so virus, okay, virus has two parts, protein code and the genetic material, okay, and the genetic material. Okay, now this protein code is made up of capsomeres, okay, capsomeres. These capsomeres become capsids. Okay, now genetic material, it could be ssRNA, dsRNA or dsDNA. Got it? Depending on plants and animals. Then we got to know viroids. Viroids are what, students? Okay, viroids are what? 1 second, sorry. Okay, not able to see the live. Okay, now able to see the live. Yes, yeah. Viroids are actually free RNA. Okay, free RNA. They do not have a protein code. Okay, no protein code. Okay, no protein code. Since they do not have a protein code, they are also going to cause problems, like they cause potato spindle disease. Potato spindle disease. Okay, now prions, okay, prions are by itself different. These people are unfolded, okay, unfolded, no, folded, okay, folded proteins. These folded proteins also cause diseases like your mad cow disease. Okay, mad cow disease or BSE. Okay, BSE. So this is also prions. This fellow has only protein, this fellow has only RNA, this virus has both of them. Okay, this is small, viroids are smaller than virus as well. Now the last topic we are left with is lichens. As I told you, we have all seen a lichen in our life, but we have never identified it. That is because like no one told us that okay, we have to identify a lichen, no. So we did not know this, but understand lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and fungal component is called as mycobiont. So algal component is called as phycobiont and fungal component is called mycobiont, which are autotrophic and heterotrophic respectively. That is, one can make their own food, another one depends on another organism for food. Okay, algae prepare food for fungi and fungi provide shelter and absorbs mineral nutrients and water for its partner. So close is the association that if one saw a lichen in nature, one would never imagine that it is two different organisms. Students, if you go to college, right, if all of you go to college, you would have seen tree. Okay, all of you would have seen a beautiful tree that is inside, very inside your college. Okay, first time you would have seen a red color tree. Wait, okay, you would have seen a beautiful tree that is inside the college, okay, where there is no pollution or anything. You will all notice that on the tree ends, okay, on the tree ends you will see some like this structure. Okay, if I draw the enlarged structure of this, you will see a very disfigured structure like this. Okay, this is a lichen. Okay, this is a lichen, that is, they inside, in the between, they will have a green color structure. Okay, here they'll have a green color structure and outside corners will be full of white structure. Okay, outside corners will be full of white structure. Why so? Because fungi is providing shelter for algae and algae is providing food for fungi. So the algal component, okay, yes, exactly, very good, pollution indicator. Okay, the algal component is called as phycobiont. Okay, phycobiont, and the fungal component is called as mycobiont. Okay, mycobiont and phycobiont. This fellow algae gives food for fungi. Fungi gives shelter for algae. Got all of you? Okay, this is the thing. And as Roshni told, these are the best pollution indicators. Pollution indicator, that is, wherever pollution is there, okay, where pollution is more, okay, lichens will be less. Lichens will hardly grow. Okay, but where pollution is less, lichens will grow. That this will be studying in industrial melanism. Okay, industrial melanism. Okay, got it? Yes. So lichens done, algae done. Like that, we have come to an end of this chapter. Okay, we have come to an end of this chapter. So today's class, what and all did we do? Okay, today's class overview, recap. Okay, recap. Today we started with fungi. Okay, we understood types. Okay, Phycomycetes, Basidiomycetes, okay, Ascomycetes, Basidio, and Deutero. Okay, Deuteromycetes. Got it? So Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Now we learned about their hyphae. We learned about their mycelium. Okay, this is the things we learned. Then we learned about asexual spores. Okay, 3 of them. 3 of them means they're not very differentiable from each other, but just we learn them. Okay, Conidia, zoospores, and sporangius. Students, the questions are going to come from this. They are not going to ask you the color of anything. They are going to ask you who has conidia, who has zoospores. Okay, all these questions only you can expect. Then sexual spores. What are they? 1, 2, 3. That is oospores, ascospores, and basidiospores. Okay, basidiospores. Got it? Basidiospores. Now we learned all that. Then we learned about each of the fungi. Okay, we learned about this. The most special character about this is they are aseptate, okay, and not branched. Okay, this one has ascocarp and it is a sac fungi. When I tell sac only, you should remember the word asci. Okay, asci, you shouldn't forget this. Then basidiocarp, we learned about the dikaryon stage. Okay, in the dikaryon stage it formed a basidium. Okay, basidium was formed. That basidium gave rise to 4 basidiocarp. Okay, basidiospores, and they were arranged into basidiocarp. So this fellow is imperfect fungi. Okay, does not believe in perfections, imperfect fungi, because we only know the, what do we know? We only know the about the asexual spores. Okay, and the asexual spore is conidia. ASM writing. Okay, asexual spore is conidia. This is what we learned. Okay, after this, all these types we learned. Then we learned about virus, viroids, and prions. Then lichens. Okay, this is what we studied. Done. This is the recap of the class. Okay, so that's it for today, students. So we have finished NCERT line by line of biological classification. Animal kingdom and plant kingdom is separate by itself. So mostly tomorrow I will be doing animal kingdom, because we are going in order. Otherwise I will let you know which chapter I'll be doing next. Okay, mostly it is going to be animal kingdom. After that we'll be done. Living world, Baswa sir will take on the same channel, so you do not have to worry. All the chapters of your 11th and 12th will be covered. So thank you so much for joining. See you all in next class. Take care and have fun. Okay, so see you all next class.